Hello, everyone, and welcome to this webinar brought to you by the Alliance for Regenerative Medicine on addressing potency assay development challenges. My name is Michael Lemke. I'm the Vice President of Science and Industry Affairs at the Alliance for Regenerative Medicine. I'm going to talk to you today about a topic that is of great interest to me, and I think many of you as well, judging by the large number of registrants that we had for the webinar today. And we have some excellent participants, and we'll talk at some length about the issues with potency assays, which I sometimes call the Achilles heel of cell and gene therapies, and also touch on what we think are some potential solutions. For an agenda today, we're going to turn over in a few moments of honor to have Dr. Peter Marks, the director of CBER, here with us, and he will make some opening remarks about the topic and the importance of what we're trying to do. And then I will spend a few minutes with some slides talking about a meeting that we call the Scientific Exchange on Potency Assays that occurred last year. And I'll highlight some of the high points from that meeting and also from the white paper that ARM produced as a result of that meeting. And then in the second half of the meeting, we'll break into a panel discussion, including some of the participants in the Scientific Exchange meeting, and we'll allow for some time at the end for some participant Q&A. Just a logistical detail: please use the Q&A feature for that as opposed to the chat. That'll make it easier for me to monitor. With that, I would like to introduce. To make some opening remarks, Dr. Peter Marks from CBER at FDA.
Hey, thanks very much, Mike. First of all, I just want to say thank you to ARM and also to ASGCT, who helped sponsor the original potency workshop. This really is a great opportunity today to discuss the outcome from that meeting. The issue of potency is one that's obviously been around for quite a while, but for our products that we're dealing with, I think it's really become something front and center now, in part because we're starting to see success. And I think it's a good thing that we're starting to really hone in on this, because now that we're actually starting to see products come through the development pathways relatively rapidly, we don't have time to work and rework versions of products, and getting a potency assay, essentially understanding critical quality attributes, getting a potency assay, and having a consistently made product really has become such a critical thing if we want to see things move rapidly towards the market. So we know that things aren't perfect, and I think we've really appreciated the feedback. Rather than being dogmatic, we want to be pragmatic and make things work here. So really look forward to dialogue, continued dialogue this area and in this area, and really thank everyone for joining today. I'll stop there.
Thank you very much, Dr. Marks, and very much appreciate your passion on this topic and also your participation in the Scientific Exchange meeting. And I think I'll open up for the benefit of those of you who may have not been familiar with what we did last year. As Dr. Marks said, ASGCT and ARM co-convened a workshop or a roundtable discussion on October 9th of 2022 on the topic of potency assays. We decided early on we wanted to have a different format for this meeting. And because of the nature of this issue, where there are some wide gaps and differences of interpretation of the guidance, we wanted to provide an environment where people could give frank feedback, but also participate in collaborative problem solving. What that necessitated is having a relatively small meeting. So you can see over here on the right, actually all of the organizations that were included in this, and typically just one individual from each of these organizations. So it was co-organized by ARM and ASGCT. FDA participated, including Dr. Marks and Denise, Gavin, and Matt Klinker, who are here with us today. We had participation from some academic experts, along with therapeutic developers. A fairly diverse group in terms of modalities. We had AAV developers represented, also CAR T developers and a TIL developer. And it was really interesting to see that in spite of the fact that these products are very different and of course have different requirements for potency assay development, there are some very common themes which emerged, which I will touch on today. We also, some of you may have seen this meeting, in the interest of transparency, we live streamed it, but we wanted to be even more transparent and open about the outcome. So after the meeting, we put together a white paper highlighting the key findings and also recommending some ways forward for the future. And then culminating up ultimately in this webinar today, where we'll tell you about the white paper, but also get some reflections from some of the participants in the meeting. So I'll touch just briefly on a few points from the white paper. And I would encourage you, if you haven't already, to read it. It's about 20 pages long. And at the bottom of the slide here, you can see a link on our website, or if you go to our website, you can just put potency assay in the search bar, or on Google, put ARM, or Alliance for Regenerative Medicine, and potency assay, and this will pop up. It's pretty easy to find. The first thing to understand about potency assays is that they have multiple very important jobs to do. And these are shown here. This is not even an exhaustive list, but I'll highlight just a few of the points. First and foremost, potency assay should be reflective of the drug's mechanism of action, therapeutic activity, or intended biological effect. And this is the case with cell and gene therapy products, and it can be particularly challenging because the mechanism of action is often not well understood at early phases. And in some cases, there are multiple mechanisms of action. Potency assay, of course, should measure the product's strength or activity. It has to meet predefined acceptance and rejection criteria, which can be challenging, both in terms of developing those criteria and also developing assays that are robust enough that they can consistently measure and acceptance and rejection criteria. Potency assays should be stability indicating, and very importantly, they should be QC friendly. So you have to validate them, and they have to be usable on a regular basis in a production environment. You may have an assay that works very well in a research setting, but if it's not robust, if it's not able to demonstrate lot-to-lot consistency, it's probably not appropriate for use in a QC environment, in a production environment. So the fact that a potency assay has to do so many things, challenging things, makes it difficult to develop one assay that will do all of these things successfully. So this leads to what we call the potency assay matrix, or the matrix approach to potency assay development. And while this is not a hard and fast requirement, it is what you most commonly see in the development of cell and gene therapies. And all of the participants in the meeting felt that the use of the matrix was appropriate, and the concept of using a matrix for potency assays is sound. However, there were some universal challenges that were identified. And I'll just give one quick example here. This is the example of a potency assay development program or matrix for an AAV vector. So typically what happens here is the developer is required to measure activity at multiple points in the biological cascade. So to measure the activity of the vector, infectivity, or its ability to deliver the transgene to the nucleus, to measure transcription and translation, and also typically to measure the function or the activity of a protein that is expressed. So you're talking here about a minimum of 4, sometimes more, distinct assays, all of which may be required for release. The challenge is that some of these assays are highly variable, and TCID 50 is one that is commonly cited as being problematic from a standpoint of measuring infectivity of AAV. Also, the question was raised, or the point was raised at the meeting, that if you are able to, in fact, measure the end of the cascade, measure that you do have activity of the protein that you're trying to express, this should only be possible if all the previous steps in the cascade have occurred. So do you really need to measure them, or what is the value in doing that? On the other hand, there are situations where these assays shown in the matrix here can be demonstrated to be highly correlative. And then the question comes up, especially in late stage, do you really need to use all of them? And what is the advantage of doing that? And can an argument be presented to, at that point, prune the potency assay matrix somehow and rely primarily on the assay or assays that are least variable and most robust for release, at least, and perhaps using other assays for characterization purposes. So this was a topic that we discussed at length in the meeting. There's more information about it in the potency assay white paper, and we'll touch on it in some of our discussion today. So before we get into the discussion with the other participants, I wanted to highlight a few recommendations, possible solutions to these issues that we identified at the Potency Assay Scientific Exchange. And it was agreed that really this is a shared responsibility, that there's a role that industry has to play, that developers has to play, and there are things that FDA needs to do as well to make progress on these issues. On the industry side, as we learn more about how to measure potency of cell and gene therapies, that will certainly help to resolve some of these issues. And a lot of that development work, a lot of the know-how, whether it's a tool or a particular assay, a multiplex potency assay developed by a technology company, or whether it's developers continuing to work on a better understanding of their own products, this will necessarily fall on industry. And one thing I think we are seeing, I can say, is that industry is starting to give a higher priority earlier on in development to paying attention to potency assays. And that's a good thing, which I think will lead to progress, and is already in some cases starting to lead to progress. So prioritizing resources, both in terms of talent and workforce, and in terms of the money that is being spent to develop potency assays early on. FDA also has a role to play here. There were needs for additional clarity on some of the guidance expressed at the meeting, perhaps modality-specific guidance, perhaps more guidance on the matrix approach and how to apply it, but also how to, the term we used was prune or cut back on the matrix approach as necessary later on in development. Also, a recurring theme that came up is regulatory communication. So, while it would be good to have additional guidance, potency assays are very product specific. So it's really not possible to be so prescriptive in guidance that you can spell out all of the requirements. And developers certainly want a degree of flexibility in the guidance. So what that really necessitates when you're talking about understanding the requirements and agreeing on the requirements for a specific product is communication, and communication of a fairly regular nature, and of a direct a nature where there are direct interactions and not just written responses. So, one of the biggest requests that came out of this meeting from the industry participants was more consistent and clear communication from FDA. So with that, I'll conclude my presentation, and I'll ask the participants in our panel discussion today to unmute yourselves and turn on your cameras. All right, well, welcome, and thank you for joining us today, and thank you for participating in the Potency Assay Scientific Exchange last October. We have here today Denise, Gavin, and Matt Klinker from FDA, and also Eliana Clark from Intellia Therapeutics and Herbert Runnels from Pfizer. Maybe before I jump into my first question, I'll ask you all just to briefly introduce yourselves. I'm sure many of the people viewing this today know you already, but if Denise, if you could maybe just introduce yourself briefly and talk about your current role at OTP.
Okay, my name is Denise Gavin. I joined the FDA around more than 20 years ago, actually. Wow. And I started out as a CMC reviewer, and then I was a branch chief for several years. And most recently, I've taken the role as director of the Office of Gene Therapy, which is in the new super office of the therapeutic products. So we look forward to the discussion today.
Thank you, Denise. Matt?
Hi, thanks. Yeah, I'm Matt Klinker, and I am currently a CMC reviewer in Cell Therapy Branch 2 in the Office of Cellular Therapies and Human Tissues. Sorry, we have all these new names and new acronyms. It's hard to remember them all. But yeah, so I've been at FDA for almost 10 years now. But I was originally I was a researcher working on potency assays for cell therapies. But I've been a CMC reviewer for almost 5 years now. And I was at the meeting in October, that the previous meeting, and got to meet everyone. So it's nice to see everyone again, and I'll pass it on.
Good to see you, Matt, and thank you for joining us today. Eliana?
Hello, everybody. I'm Eliana Clark. I'm the Chief Technical Officer at Intellia Therapeutics. I've been with Intellia for about 3.5 years. Intellia Therapeutics is a genome editing company. I was at the workshop that we had in October, and lots of things that we exchanged and discussed and learned, and the white paper resulted from it. I've, in my career, I've worked with proteins, small molecules, oligonucleotides, etc. So a variety of experiences in my past that have prepared me to actually come into this field and bring those experiences and see how we can make a difference. And I think, as you said, Mike and Peter and all of us, potency assays are really important for what we do today.
Thank you. And last but not least, Herb.
Hi, Herb Reynolds. I've been at Pfizer here in St. Louis, Missouri for a little over 20 years. And my role that's relevant to this is I'm the analytical sponsor for the AAV products and their discussions and interactions with the regulatory groups around the world.
Thank you, Herb. So let's start out actually with just a quick logistical thing before I jump into the first question. I did say at the beginning that the audience could use the Q&A, but for some reason I'm not seeing it on my menu bar. So as we're talking here, if you're in the audience and you think of questions, feel free to use the chat function, and I will keep an eye on that, and we'll come to some Q&A at the end. But I'll start out, and each of you has somewhat, all very knowledgeable and experienced with respect to potency assays, but you have somewhat of a different background and different roles, of course. So I think it would be great if you could share what was your takeaway from last October's Potency Assay Scientific Exchange. Maybe what was your main takeaway or the most important one for you? And was there any, were there one or two particular learnings that you were able to take away and maybe take back to your colleagues at your respective organizations? And maybe I'll start with Denise.
Okay, yeah, so thinking about the meeting last year, I think the main thing that I took home from that was that there's still challenges with potency testing. And the second take-home message I had is that there's misunderstandings about the guidance and what we're actually thinking about when we wrote the potency guidance in 2011. And I tried to explain some of those thinkings at the meeting. I still think there may be some misunderstanding about the cascade versus a matrix, and maybe we can talk about that at some point further. I also took home that there's a lot of this misunderstanding is probably due to miscommunications, either through the agency, through the misunderstanding of the guidance. And I think that as the agency, we started thinking about a lot of our processes and how we could improve them for increasing communication. And one of the things that came out of the PDUFA agreement was type D meetings. And I think those type D meeting, that format may be excellently designed and to facilitate potency discussions. So we can talk about that more at another time, or even today if it comes up again. So thanks. Those are my main take homes.
Yeah, I think I'll come back to the point about the type D meeting if you don't mind, because I know that's something you raised that day. It was the first time that I had heard about it at the meeting that we held last October, and I think it's a very interesting idea. Matt, would you like to go next and share?
Sure. So, for a one-day meeting, I think we had a good discussion about the challenges of how to develop these potency assays. But we've been talking about this for a long time, and in one meeting, we're not going to solve all those issues. So, for me, the real takeaway really was about communication, and I guess the relationship between regulators and developers. There's an inherent just frustration there, I'm sure, that is unavoidable. It's the nature of the relationship. And so, and some of that's just from the nature of the relationship, but I guess my takeaway was that we could do better in communicating. We should do better in how we communicate, at an individual level, when we're communicating directly to developers. And so that's something I think we can work on. And that's what I took away from that workshop. And it's something I've been thinking about and working on my personal communications to sponsors is trying to be more clear about what we want, what we expect, and maybe also being clear on what things are recommendations and what things are not just recommendations. There are things you actually should or have to do, and so I think that's that room for that communication between between the both sides of that here. I think is that was the thing that I think that made the most progress on in that meeting.
Yeah, it's interesting that you highlight communication, and I completely agree. And you use the word frustration that can occur, when there's miscommunication, perhaps between developers and FDA, and that actually came to the surface a little bit at this meeting. So, I
it got a little spicy. It got a little spicy, but yeah, it's the nature of it. Yeah.
Commend all of you and the other participants for working through that in a constructive way. Eliana, would you like to go next?
Okay, sure. One thing that I learned is that we're not alone. I think the challenges with potency assays and understanding the 2011 potency guidance seems to be something that happens to pretty much every company, every one of the developers that participated, and even some of the academic institutions that were there. So that was pretty clear at the meeting. So we're not alone in having sometimes difficulty understanding what the requirements are. I think Denise mentioned, and it was a good topic of conversation, that the guidance was written with some degree of flexibility because the cell and gene therapy space was just starting to be developed. Now it's exploding, and perhaps that flexibility that was well intended, as you said, Denise, was actually have resulted in ambiguity and a bit of confusion. And that was something that, again, happened pretty much, we all agree across the board. What I learned that I took back to my company is the need for us to have more clarity. When we put in any regulatory filing or any meeting request, when we speak about a potency assay, not just describe this as a potency assay, but why? Where does it come from? Why did we come up with this assay? Why does it represent the mechanism of action? Why do we believe it's the most appropriate assay? So put a little more information in there than just a description of what the assay that we intend to do, to run is. The other thing is to start early and to actually ask questions earlier. I think it was mentioned in the meeting, use your interact meetings, use your pre-IND meetings, even though the potency assays, the matrix and some of the more complex potency assays are required later in development and not for a phase 1. But unfortunately, these assays take quite some time to develop, and learning early on them is important. So asking those questions early on in those early meetings about what that potency assay might look like is something that I took as a key message. And then agree with you, Mike, the type D meetings as an opportunity to actually talk about, it's a focus meeting, and we can talk about that later, but it's a focus meeting. and we can talk about potency assays early on with FDA. I think was also a good takeaway.
Yeah, and I appreciate your comment about developers sharing information about their thinking and how they developed the potency assay and methods. And that was a big takeaway for me from your comments that you made, Denise, as well. You know, we talk about communication and clarity of communication from FDA, but it's a two-way street. And, you know, developers have to communicate what their intent is as well and what their thinking is behind the potency assay or assays that they're developing. Herb, could you share your key takeaway or takeaways from the meeting?
Yeah, sure. So I saw it as a great candid and informal environment. And what I mean by informal, it wasn't focused on trying to advance a product, you know. And so what I learned from that is that the FDA is actually listening. I had a greater appreciation for that. They're discussing things internally, and they're trying to do group reviews to make sure consistency is in play. And I got a better understanding for what they're experiencing. And maybe these are lack of early investment by from some sponsors, or maybe some sponsors ignoring advice or misunderstanding the wording of the advice that they use. And I think we're in it together. So for me to understand that a little bit better was very valuable. I also understood what some other companies, maybe like the CRISPR companies with all their different components, what they're being asked and their struggles there. And I think one of the takeaways, and we'll get to that more later, is we've now written a really detailed, like type D briefing document with a lot of method details. We haven't submitted yet. They still have that to look forward to. But a lot of that is an outcome of this meeting and from things that Matt and Denise said at that. So I enjoyed the informalness, not tied to a project, but tied to industry and regulatory interaction.
Thanks, Herb. Yeah, it sounds like it was a very constructive day for you. So I'm going to ask a question of Denise and Matt now, and you can just jump in, whoever feels most comfortable with it, but drilling down a little bit. For a gene therapy vector, how might an expression assay be used in lieu of an activity assay or in place of an activity assay to meet potency release testing requirements? And if you could just comment on how the agency's thinking is evolving on this topic.
I can take that, since I reviewed potency tests for gene therapy vectors for 15 years or so. So I can start. Matt might have something. So generally, we do allow expression assays. So you mentioned that there's this cascade, and you start with infection all the way to the activity of the product, and that you're required to measure all those things. And I think there is a misunderstanding there as far as that goes, that whether you have to measure every single step as a matrix. So the matrix was envisioned a little differently to try to use the other testing that you have in place to support the potency, because you may not be able to develop a good potency test. So that's just something I wanted to, maybe we can talk about that more. But as far as gene expression, so we do allow that during early phase development, especially when the risk of having a product with less potency is less, say early phase, as the risk of having a product that's less potent becomes more critical, say in a late phase study, then our expectations increase. So the real function of a potency test is to show that the product is capable of mediating the intended therapeutic effect, right? And the therapeutic effect is generally described by the function of the protein expressed in the vector. So that's why we're looking for the functional activity of the protein expressed from the vector. But early on, we will allow some flexibility to look at the gene expression, because that is one part of the product quality attribute that's necessary. So like without expression, you're never going to get the therapeutic effect, right? So we understand that. So if you were to go forward with just gene expression, I think that what you need to really do is have more controls in place. So because the gene expression doesn't really tell you that the product is capable of the intended effect. It's part of the effect, but not the whole thing. So what you need are other controls in place. So you may need to fully characterize other aspects of the product, which is what the matrix was more intended to do, which is to look at some of the other product characteristics to support potency. And then you also need to make sure you have a lot of controls in place for manufacturing, for the other tests, that they're well controlled. And the gene expression test that you have needs to be precise, needs to be accurate, needs to be very well controlled and designed. So there's you need to get to that level where that test is actually meaningful to us as well. But I think in the end, if you can have enough assurance, you can assure us that the product is capable of the intended effect, we may look at gene expression as part of potency testing.
Herb or Eliana, is there anything you want to reflect on there? Herb, I know this is a topic you've opined on from time to time.
Yeah, and I've discussed this with Denise extensively, and even at that meeting, and we don't need to rehash that. I think, one of the aspects that we would argue is that in the case of AAV and many of the other gene therapies, the product is a nucleic acid. So its biological effect is expression. Nothing we do in vitro mimics how the body will make a protein. There's nothing in our manufacturing process that affects the protein, transgene protein. So I think from our perspective and where we would like to, continue to work with the FDA is a greater understanding on if we can control that expression assay and certainly support activity with characterization tests, maybe in vivo models, maybe in vitro assays, but just not on release if it's not possible. Sometimes it's impossible to generate a functional activity method for certain proteins based on their functions. And so I think that's where we see that. So I see what Denise says as an opportunity to further discuss that. And I hear what she says about you got to have good controls and controls around that. But I think for some products, we're going to need that flexibility as we're manufacturing a nucleic acid product and not a protein. We can still show activity. And even in the clinic, you can show clinical efficacy, or you can insist upon that, for a program like that. So I think it's a good discussion. I really enjoyed that day with Denise. And again, it was more informal than us discussing particular projects. And
Yeah, so it sounds like I wouldn't say you're in complete agreement, but I heard some common things. Use of characterization and characterization testing in the concept of the matrix to support the potency assay and perhaps other types of release tests. But also, I guess I would say you can't test in quality at the end of the day, right? So it's a great point that you both brought up about manufacturing controls really being a supportive and important part of quality.
Yeah, so I'm not disputing the need for activity. I'm just saying for some cases, activity might not need to be the release test based on that whole control strategy that you put in place.
Yeah.
Right.
Yeah, you probably.
And I think that's what I.
Go ahead.
Oh, go ahead. No, go ahead. Then
no, I'd rather hear what you have to say.
I was just what I was going to say is I think that part of the conversation had to do with, at the end of the day, we end up having all these assays. And if we could have more characterization, understand our product, and many of these assays in the cascade that we were talking about are characterization until we find the one assay that really, as you say, Denise, this is not a matrix. The cascade is not the matrix that the guidance was speaking to. And so if we could find the one assay in the cascade that is actually the one that we want to use for release, for etc., stability, and then have the other ones as comparability, as characterization assays that can be used also for comparability, etc. I think that was also where the conversation went, because it ends up being that we end up qualifying and validating multiple assays that are all in the same, they're correlated.
Right. Good point.
Yeah.
So
But
maybe
I
I'll.
interrupted you, Denise, so I'm sorry.
good.
Oh,
Is
I
there
was
some-
just
if you
going to
wanted
say,
to
I was
add.
just going to talk about the need for correlation if you're looking at other things that don't directly measure the activity of the, the therapeutic effect of the product. Gene expression doesn't measure the therapeutic effect. So if you're not directly measuring the therapeutic effect, you need to show some kind of correlation to that activity. That's how you could probably, is going to be what's necessary to use gene expression. So.
And would that be correlation with clinical
Well, it's
outcomes,
difficult.
or are there other ways to do that?
I mean, that's a little more difficult just because of the amount of data you would need. And, our trials aren't very big in cell and gene therapy. So it's difficult to correlate to clinical, but you can have preclinical, you can have other assays that you have developed to show activity. And if the product is working, you're going to know. And so eventually you can show some sort of correlation to the clinical, but it's very difficult to get enough data to do a clinical correlation just because of the size of our trials and the variability of the patient population. And, there's a number of caveats to that sort of approach, as you can imagine.
Yeah, thanks for
Mike,
that.
I wonder if a spinoff might be a working group that tries to draft what that data package would look like and what aspects, and see if we can co-create a roadmap for what could be successful, and because again, I'm not saying to eliminate assays or eliminate understanding. I'm just like, what is the CMC release assay, which is a higher bar on every lot testing versus what you apply to certain assays or incomparability exercises when you make a process change. And so maybe that's a potential spinoff for the future, that we could have a working group, what have you, to try to design a roadmap of data that might be doable and acceptable.
Yeah, that's an interesting.
Yeah, I was just going to say it may not be the same for every vector, right? So we're talking about gene
Right,
therapy vectors in general. Some are much more stable than others. So the sequence itself may be different. So, just gene expression may be okay for some because the sequence is stable, may not be the same for a different product that is much more variable. So it all depends on the product that we're looking at. So sometimes these conversations do need to be in the context of product development.
Right. But perhaps, your idea about a working group is an interesting one, Herb, and perhaps it could, there could be some subgroups within that look at particular examples or archetypes that could be useful. So I want to circle back to something that came up earlier, and the topic is communication with FDA. And if a developer has specific questions about their potency assay or potency assay development strategy, Denise, what are the best ways to interact with FDA? And you mentioned a Type D meeting earlier. Maybe you could say a little bit more about that.
Yeah, so that's a new PDUFA requirement that we introduced a new Type D meeting. There is also Interact now as a PDUFA meeting. So those are both meetings where you can ask specific questions that are particular for your program. Both of those require the package come together with the requests so that we can see whether you have questions that fit that meeting type and whether you have enough data to support the question. Because that's sometimes what happens is we get questions about potency where we don't have enough data to actually give productive comments. And so what you get are general comments that lead to more confusion. So then you go back and forth until we finally get something that's agreeable to both. But at this time, we've wasted a lot of time. So I think these are having the meeting package so we can look at it right away before we start, and then we can know if you have enough information there. So those two meeting types, Interacts and Type Ds, allow us to do that. There's also, you can ask questions in your pre-INDs. You can also submit information to your file as an amendment on your cover letter. You ask a question about the information submitted, and we will respond to those questions in the context of an IND amendment. There's also milestone meetings that you have. A lot of these products have RMAT and breakthrough. You can ask questions about potency at those meetings as well. So there's a lot of mechanisms to discuss potency with the agency. Did you have anything to add to that, Matt?
No, I think I would just say that if you mentioned all the different types of meetings that are available, and really, I think the best time to ask questions is when you have them, and when they come up. So, any chance you have a meeting coming up, and you have questions about your potency assay, put them in there, and we'll take a look at them. But yeah, I think the type D, that'll be interesting to see how that goes. I think it's, as I understand, it's more directed, very more focused type of meeting. So that might be. We'll see how it goes. I've only had one of them, so I don't have a lot of experience with it yet. So, but we'll see how it goes. But yeah, I think just when you have questions, there's plenty, those opportunities. Just every meeting you have to ask us questions about potency.
And Herb, you mentioned. Go ahead, please.
Sorry. So a caveat that can be within a company or a sponsor that your timing of approaching the FDA might be tied to something internal, like tied to a conversation clinical wants to have. So you may not be able to get to the FDA to ask those questions. Like your type D meeting, there's like 2 questions. The other question might not be ready. And the converse has also been true in our experience, where the FDA has said at times, we don't want to talk about X until we've resolved Y. And so I think one thing I'm hearing and I've experienced is that potency assay is a huge log jam. And, not that I'm trying to dump another meeting on you, but I would welcome, when we filed our IND, if the FDA would write back, we want to have a 30-minute telecon to discuss your potency strategy, and just have that. So it's disconnected from anything else and being tied to anything else. And I would love to lay that out and get that input. And I realize that's resource constraints, you're expanding, et cetera. But I just want to point out that the list of meetings, there's a lot of opportunities, but they're frequently tied to things like non-clinical, safety, and clinical that are outside of CMC's control. And it's actually a reality that becomes a logistical concern. But the minute the FDA says they want to talk about it now, then I can engage you right away. And so anyway, if that makes any sense. And
frankly, that sounds like a good, from my perspective as a reviewer, that sounds like a good idea. Just a nice, a 30-minute, maybe informal telecon to discuss that. Something. You can always ask for informal stuff too if you want. That doesn't mean we're always going to, due to time constraints, we might not always grant it. But yeah, honestly, that sounds like something that might be helpful. I don't know how feasible that is for us to do on a every IND basis.
Right, I understand. I'm learning your constraints more and more as time goes on, and thank you.
Right.
Let me add that.
Yeah, I think informal communication can be very valuable and important, but obviously you need the resources and the time to do it. And I guess the right I would say understanding and guardrails need to be in place to do that as well. But I think with I know one thing that I'm hoping is with the OTP getting additional resources and the super office build out, there'll be we can look forward to more opportunities for less formal communications. Eliana, is there anything you wanted to add on the topic of ways to interact with FDA or the Type D meeting specifically? Have you had any experience with that yet?
Yes, and actually, what I wanted to comment is we at the meeting when we met in October, we talked about the direct interactions, and we understood that what was happening in the country over the past several years with the pandemic, et cetera, there were challenges at the agency. And I think we agree that those direct interactions are incredibly valuable. So yeah, I think we have, taken advantage of a Type D meeting request. We'll see how it goes. So it might be a first for us, and it may end up being a first for as you said, Matt, you only had one, so maybe it might be the first for the folks that might participate from FDA in that Type D meeting.
Yeah, we've had several. I also wanted to throw out there that there's the CMC Development Readiness pilot that we are taking applications for, and that pilot, you can use those meetings if you get into that pilot. You do have to have RMAT or breakthrough for a cell or gene therapy. But if you get into the pilot, you can use those meetings to discuss potency issues, development issues along those lines as well. So that's one more avenue that I probably should mention, since it's a new program.
Yeah, I'm glad you brought that up. And since you did, maybe I can just ask. So applications for that program are still open? And is there information on the FDA's website about that?
Yeah, it's on the FDA website. We started taking applications in April, and we'll continue to take them till the program is full, and then we'll start again next year.
Okay. And is there kind of a timeline and vision as to when applicants will find out if they've been accepted into the program, so to speak, if that's the right terminology?
I think we'll evaluate the applications quarterly, and then we generally get back to people right away if they qualify or do not qualify.
Okay, so it sounds like it's kind of a running process until the program is full.
Yes.
Okay, great. Thank you. Yeah, thanks for bringing that up. That's very interesting and informative. So I want to come with my next question to the industry, to Eliana and Herb, and then maybe I'll ask for Denise and Matt's perspective on this topic as well. But what you hinted at some of these things, but what have you seen from your organization or maybe outside of your organizations that's changed in terms of approaches to potency assay development since we met in October? And what do you think still needs to change? And a related question I have is, do you think there's a role for information sharing among developers to help overcome some of these issues? Eliana, maybe I see you nodding, so I'll ask you to start.
Yeah, maybe I start with your last statement. I think that there's absolutely an opportunity where and we can get together as developers and start talking about the roadmap. I think Herb mentioned this. During the meeting in October, we talked a little bit about having like almost decision trees, depending on the type of products that we are developing. I think the challenge that we have is that in the cell and gene therapy space, there's so many different modalities that one guidance doesn't fit all. And if we want to cover all the different modalities, it becomes a very high level guidance. And I think we have an opportunity as developers to work with organizations like ARM to come up with a roadmap or a decision tree that may allow us to then determine what are the right, at least a guidance of where we go with the potency assays. I see more. We also talked about the fact that we don't typically present at conferences our potency assays or our potency assay strategy. I see more presentations at conferences of other areas of what we work on from a development point of view. So I think potency assays will come because I see more disclosures of how we are doing our product development, especially in like the process and analytical development space. So I envision that in the future there will be more opportunities for companies to actually talk about how we have developed potency assays. I don't think we're there yet. We still may have offline conversations with our colleagues, but not really an open forum conversation on potency from the developer side.
Yeah, thank you for that, Herb. What's your perspective on information sharing, and what have there been positive changes since the meeting, or what do you think developers still need to do to improve the situation?
So that was discussed, I think, very well at the meeting, and that also got kind of tense there, and I think that's good. And I think companies and academics need to share more on that. And I think internally, we have plans to share on specific potency assay aspects, but it's not till approval. And but that's an important thing for us to do. And so it needs to reach into our resource constraints. Just like the FDA has many projects, we have many different things we have to advance. So publications might not necessarily be our first priority, but I heard in that meeting that is a huge value to the organization. And Pfizer has published multiple things on AAVs, including just an isoelectric paper last week that talks about how to measure protein modifications, And so I think you're going to see more and more in that. And I think last year Pfizer did presentations about their different potency assay correlations where they actually named the programs, whereas before they were generic. So Pfizer is evolving, as we move through these steps to share more of our stuff. And I think Spark in the past and Biogen have done a fabulous job of sharing details of assays and stuff. And I really think that's valuable. And so I heard that criticism, and we've discussed it internally, and it's more of a priority for us to get manuscripts out as opposed to just posters or presentations.
That's really fantastic to hear, Herb. But any thought I have, it sounds like you're really working at this issue and passionate about it. And any advice on how to deal with internal legal people? Do you take them out for a drink, or how do you convince the lawyers that this is the right thing? This is the right thing to do.
Yeah, that's probably company specific, but in Pfizer, usually analytics aren't, an IP thing. But some of the analytics are key, product specific or competitive. So they might wait for approval of the license. But actually, Pfizer has a very robust policy of trying to help others as well. We feel we're in it together. There's a whole health equity charge that Pfizer has in their culture, and I think this plays into that.
Yeah, it's great to hear. I don't know, Denise or Matt, any comments you want to make? I know the agency is restricted in some ways about the ways that it can share information. Certainly, you can't share confidential information from a sponsor, but have you seen more information sharing in public venues from industry, and how important is that, do you think?
I don't think I think the time since that meeting was relatively short. So from what I've seen, I haven't seen any trend really. But as you said, we don't really have a role to play in that idea of sharing information between developers. But I guess I could say it sounds like a good idea and helps the industry. So go for it, guys. But that's about the extent of my contribution on that.
Yeah, I just wanted to add that I've been involved in a couple different bioassay meetings for years, and Matt took advantage of, Matt presented at one of those last year at the CAS meeting. But there's a number of meetings out there where people do just talk about bioassays and just talk about potency testing and challenges. And those are a lot of times at those meetings, you'll run into developers who have years and years of experience in very particular aspects of bioassay development. So those might be helpful to industry as well to participate in them. There's quite a few of those meetings out there, so I think that might be helpful. And if asked to present, maybe it'd be helpful if you share whatever you can at those meetings as well.
Yeah, I will second that. And you see some really good assays. So it might be a little scary for people that are trying to develop their own, but at those meetings there's some really experienced and really good assays, and so it's a great resource for learning. Yeah,
that's a great point, Denise. And maybe we can talk a little offline about that at some point. I mean, we're happy at ARM to promote the efforts of others that are moving this issue forward, or present there or partner up as appropriate. So I want to allow some time for questions from the audience. I have one eager participant who submitted questions in advance, but also Savannah, I'm not seeing anything in the chat. I don't know if I have to believe there must be some questions out there. So maybe if you could let me know if you're seeing anything at your end. I see a couple of hands raised. But what I'd like to do is take the questions in written form in the chat if possible. But in the meantime, technical question actually for Denise or Matt. What's the typical percentage of variability that is observed with potency assays? And are there any acceptance criteria around that, around limits of variability or a coefficient of variation?
Excellent question.
Well, I guess I'll go first, and Denise can tell me if I'm wrong. But so I think it really depends on the assay. You know, if we're talking about potency assays, we're talking about an extremely broad category of assays, and some can measure something very precisely, and some simply cannot. And so it's very hard to give a specific, you know, CV or percent CV and say that that's acceptable. So it depends on the assay. It depends on you know, and it also depends, you know, if you have a matrix of assays. That, you know, because it might be acceptable to have a little more variability on one assay if you have another assay that measures something maybe related to what you're measuring in the other assay. But if that second assay measures it very, very precisely, gives you better assurance, that, you know, that might, you know, that has to, you know, I guess we kind of would look at the entire package. And so just giving one specific number is not going to be helpful in most contexts here.
Thanks. So there's a few questions about when it's required to have, at what stage of development is it required to have release criteria for a potency assay, and when does a potency assay have to be fully validated? I think there might be some straightforward answers here, but it's an important question. So maybe if Denise or Matt, if you could comment on in what clinical phase does FDA expect developers to have a validated potency assay?
Validated would be by the licensure when you come in for licensure. We do expect you to have an assay qualified for phase 3 or studies that will be used to support licensure. So a lot of times early phase trials are used to support licensure. So if you're going to use that data in your clinical efficacy package, you probably need a qualified potency test by then.
Right. And that's often the case with these therapies, I would think, or it's not uncommon, at least.
Not uncommon. Mm-hmm.
And when are you looking for release criteria for a potency assay? And I'll circle back to you, Herb.
As soon as you can get it, we would probably want you to have some limits for the pivotal study. A lot of times early on, it's report results. You know, we ask for it, but we will accept report results for a phase 1 trial because you're just collecting the information.
Got it. Herb, you wanted to add something?
Yeah, I know. I wanted to ask Denise a clarifying question. She says that early phase trials are used as part of the efficacy package for approval. You probably need like your qualified assay for the early phase trials, and people interpret that as a phase 3 level assay. But wouldn't it also be acceptable that when you have that phase 3 level assay to go back and test your retains from those early lots as part of that bridging exercise if you didn't have the assay in place at phase 1?
Well, what I'm saying is that there are trials where the patient population is so small that the trial phase 1 is considered a pivotal trial, and they use that data to support the licensure. So at that point, they need to have some sort of assurance of potency at phase 1.
Okay, so there might not be a bigger phase 3 later is what you're describing. Okay, thank you.
Yes, that's right.
Matt, I have one for you. Denise went into a fair amount of detail with the gene therapy example, but there's a question from the audience. I think you're in the cell therapy and tissue engineering world, if I'm right. Could you maybe talk a little bit about a potency consideration from that arena, or maybe give us an example there without being more specific than you can be?
So just an example. I guess I don't understand that question. You wanted me to talk about like a specific product or something? I didn't understand what you're looking for.
Yeah, the question is, since you touched on gene therapy example, can you comment on regenerative product potency?
I mean that's I think that's almost too general or too vague for me to really give anything helpful there. So sorry, but yeah, sorry.
Okay, so maybe I would encourage the questioner to check out some of the town halls. That's a good question. That'd be a kind of question you could ask maybe at a town hall, but it's just too general really for us to give anything meaningful back. Great. So we're about out of time. So I know there's a lot of questions from the audience, but appreciate your hanging in there and your understanding and our not being able to get to everyone. I just want to close by thanking all of the participants and encourage everyone to take a look at ARM's potency assay white paper, but also be aware of some of the other meetings that are going on out there that Denise mentioned, that are specifically analytically focused. And with that, wish everyone a good, hopefully fun, long weekend, and looking forward to connecting with you all in the future.
Thank
Thanks
you.
for having us.
Helpful.
Thanks very much.
Thank you.
Thanks.